Dear colleagues, let me briefly introduce you to a research topic that we are focused on in our group. I called my lecture Chasing the Retrovirus because we are focused on study of retroviruses. Why we study viruses? Viruses are very interesting agents that are quite simple. They consist of protein shell covering and protecting their genomic nucleic acid. And they are usually causative agents of sometimes very severe life-threatening diseases. Thus, it's quite important to study them into details and to try to design some approaches against them, how to fight them. You can decide either for immunization against viral diseases, or you can try to design some antivirals and approaches that are based on very detailed knowledge of the virus life cycle. And that's what we are focused on. We picked up the group of retroviruses, where belongs, for example, causative agent of HIV, of AIDS. It's a HIV virus. And since we can't study HIV and work with live HIV in here, we study or we used as a model virus monkey form of this retrovirus. Here you can see the life cycle of this virus, and we are focused on the formation of virus particle, mostly from this life cycle. What's a quite interesting property of retroviral proteins or structural proteins that form this virus shell, this protein particle, is that they are able to self-assemble. So if you prepare the protein in sufficient concentration and under certain conditions, you can force them to self-assemble and to make virus particles by themselves. So we prepared and purified enough proteins, structural proteins of Mason-Pfizer monkey virus, that's MPMV, and also other retroviruses, and optimize the conditions of this assembly and assemble these particles in vitro and study the influences of protein concentrations, added nucleic acid, pH, and other chemical and physical factors on this assembly process because we wanted to understand this process into very much details. Here you can see our results. We were able, under certain conditions, to force the protein to make spherical particles, about 70-80 nanometers in size. And under certain conditions, also, we were able to prepare the tubular particles or multilayer particles or particles of various morphologies. By further optimization, we developed a highly efficient system of formation, such as small vesicles, small particles, protein particles that we can modify their surface, or and also we can incorporate into them polyanions or various nucleic acids. So here you can see these protein particles by transmission electron microscope, and we are able to prepare huge amounts of these particles. Thus, we were also thinking how to use this system, not only to understand how the protein interacts and how to prevent their interaction and inhibit this process, but also how to use these particles. So we modified their surface by short peptides and use them for immunization. And because the virus particle itself is highly immunogenic, we also made antibodies against the modifications of the surface. Another approach was to modify these particles by another short peptides that serve as a ligands to some certain receptors present on selected cells, and we use this system as a delivery system because we incorporate it inside silencing ribonucleic acids. Here you can see these particles again by electron microscope. But it was necessary to know the structure, ideally 3D structure of virus particle, to see how the proteins are organized, how they interact, and so on. For that, we used firstly transmission electron microscopy and atomic force microscopy. Here you can see obtained pictures, but that was not enough. And we moved further and asked the colleagues from EMBL in Heidelberg who were trying to solve 3D structures of HIV and RSV, other retroviruses. And in collaboration, we solved also the structures of MPMV and compared these 3D structures of these retroviruses. We found that not as other viruses that usually have some regular structures of their virus protein shells. Retroviruses are not complete spheres, but incomplete spheres where these hexagonal organizations of proteins are full of holes, disruptions, and irregularities. And further, we discovered that it has probably also the reason for their life cycle. By using higher resolution cryo-EM, we solved the structures of our MPMV particles. Here you can see the protein organizations within the tubular particle. Here is the hexamer of proteins, and this is the side view where you can see by these electron densities, it's in gray, how the proteins could be organized. And if you know the structures of, I mean, for example, from NMR or from X-rays, structures of that certain proteins, you can fit them into these cryo-EM maps, and then you can see where are the protein-protein interaction interfaces that you can inhibit, and which amino you can predict which amino acids would be important for these interactions. Thus, what are we focused on right now is the point mutations of amino acids in these virus structure proteins, and trying to see how it influences the assembly of particles, not only in vitro system, in the Eppendorf tube, but also in infected cells, how it affects the virus assembly, release, maturation, infectivity, and so on. So here you can see the hexamer of these proteins, here individual amino acids that we predicted could be important for this life cycle. For example, here it was a point mutation that changed the place where the virus assembles inside of cell. These are infected cells, and by the green you can see where the virus particles are formed. Here is a list of used methods that we use for our research. So if you see some methods that you would be interested in, you can ask us. Some of them are in collaboration with other groups, not only from here, from UCT, but also abroad. So these are methods of DNA constructions, mutagenesis, protein purification, virus particle formation, but also cell tissue and mammalian tissue culture cells, and observing the whole life cycle of these viruses in direct environment. And here I would like to thank my colleagues, not only from UCT Prague, where we have a close collaboration with central laboratories and with Department of Biotechnology, but also some groups abroad. Thanks for the attention.